Hello, good morning, everyone. My name is Stephen Wu from University of South Dakota. Today, I'm going to be presenting my research project, all-in-one nanoparticle for breast cancer treatment based on phototherapy and chemodynamic therapy. As we know, breast cancer is a number one cancer in female, which causing a lot of health losses and mortality. So it is important to develop new methods for the treatment of breast cancer. And in this project, we proposed to develop a new nanomaterial that can kill cancer cells, can kill those, breast cancer cells in vitro and in vivo through different mechanisms. So as you can see in this scheme experimental design, we have we have developed a nanoparticles which composed by a semiconducting polymer, which we call PCPD-DPT, and then coupled with ions, which is a chemodynamic agent, which can be used to cure cancer through ferroptosis. And then we coupled with PSMA-CoH, which is another amphiphilic polymer. By combining those three different materials together, we prepared a nanoparticles we called P dots, docked with ions. So after the synthesis of the nanomaterials, we modify the nanomaterial with another enzyme, which is glucose oxidase. So as you can see on the surface of the nanoparticles, the glucose oxidase will be modified through chemical conjugation. So with this nanoparticles, there are several functions to cure cancer. The first function is glucose oxidase, which was modified onto the surface. As you can see, in the presence of the glucose oxidase, it will consuming a lot of glucose and convert them into hydrogen peroxide. So in this step, the glucose will be dramatically consumed, which will cause starvation for the tumor cells that may be used for the cancer treatment. However, moreover, the generated hydrogen peroxide in the tumor sites will be reacted with the ions in the nanoparticles and the acidic tumor environment, which will causing the generation of the hydroxy radical. So this hydroxy radical is a strong oxidant, which can be used to destroy like DNA, RNA, proteins in the cells, which will cause in the ferroptosis. So this is a second mechanism. This is a second mechanism that can be used to kill cancer. And the third mechanism is called photothermal therapy and the photodynamic therapy, which we combine them as phototherapy. So the photodynamic therapy is caused by the semiconducting polymer, PCPD-DPT, in the nanoparticles. And the PDT, which is photodynamic therapy, is also coming from the presence of the PCPD-DPT. And the laser irradiation, the PCPD-DPT will convert the laser energy and causing the temperature increasing as well as the generation of the singulated oxygen. So both high temperature and singulated oxygen will kill the cancer cells at the same time. So as you can see in this process, the starvation is one method for cure cancer. The second one is chemodynamic therapy. Third one is photodynamic therapy, and the last one is photodynamic therapy. So with this four therapeutic strategies together in only one nanoparticles, this can be used for breast cancer treatment efficiently. So in order to improve our concept and the design, we prepared the nanoparticles and characterize them with different technologies. So the first two is a TEM image of the nanoparticles. As you can see, the nanoparticles is around 100 nanometers after modification of the glucose oxide. So this is a size distribution. and it is around 100 nanometers, which is very good for targeting tumor tissues through the passive targeting effect. And then this one shows under near infrared irradiation, the singular oxygen generation was detected by the SOSG. So as you can see, under the laser irradiation, the nanoparticles can dramatically increase the fluorescence of the SOSG, which is a probe of the singular oxygen. So we concluded that these nanoparticles can be used for their photodynamic therapy and the laser irradiation. At the same time, we observed the temperature change. As you can see, with the laser power increases, the temperature was dramatically increased. So the photothermal therapy is another effect that can be fulfilled by nanoparticles and the irradiation. And also, the chemodynamic therapy effect was characterized by the TMB absorbance. So as you can see, with the increase of the glucose oxides, with the increase of the concentration of the glucose, the nanoparticles can convert the glucose into hydrogen peroxide, which will react with ions generating the hydroxy radicals. And the TMB, which is a probe of the hydroxy radical, and in the presence of the hydroxy radical, the TMB will change the color from colorless to little blue, and we can see the absorbance was dramatically increased with the increase of the concentration of glucose. So therefore, we can conclude that these PDOTs, these nanomaterials can be used for converting glucose into hydroxy radicals, which can be used for the chemodynamic therapy. So with this, the characterization in the solution, we investigate the effect of our nanomaterials in vitro. So as you can see here, A is a fluorescence image of our nanoparticles incubated with breast cancer cells, which is MCF7 cells. And as you can see, with the time increases, most of the nanoparticles will enter their tumor cells, and we can observe that from the red fluorescence, which is coming from the materials. And also, we investigated the cell variability of different groups using, the MTT assay. And as you can see here, with the nanoparticles and the laser irradiation, the cells availability dramatically decreased, which indicating that the nanoparticles can kill the cancer cells after the laser irradiation. And here C is a proof of the generation of the reactive oxygen species, which including hydroxy radicals as well as singular oxygen by photodynamic therapy. So under the laser irradiation, as you can see, most of the cells was dead by the indicated by the red fluorescence of PI. So we also figure out that these nanoparticles can make the cells go through ferroptosis, which was causing by the ions in the presence of the hydrogen peroxide. So GPX4 is a standard, GPX4 is an important protein in the pathway of the ferroptosis. So as you can see here, in our nanoparticles treated with tumor cells, the GPX4 was dramatically decreases, but in the presence of the inhibitor of ferroptosis, which is FER1, the level of the GPX4 restored a little bit. So that indicates the nanoparticles were causing the cell death through ferroptosis as well. So lastly, we investigated the cytotoxicity of our nanoparticles to the breast cancer cells as well as another tumor cells, and we can see that in the presence of the nanoparticles, our cells viability dramatically decreased. However, with the incubation of the inhibitor of the ferroptosis, the cells viability go back to their about 80%, which indicating that the chemodynamic therapy plays a very important role in this process. So with the exciting results in vitro, we applied the nanoparticles into the tumor, which we constructed a tumor model by the MCF7 cell line. So as we can see here, we established a tumor model on the mouse under mouse skin using the MCF7 cell lines, and then around like uh, 6 weeks old of the mice after the tumor was established, we inject the nanoparticles and irradiate the tumor with a laser, and then we observe what will happen in the tumor, whether they cause a tumor suppression or not. So first of all, we detect the temperature change after the laser irradiation. So as you can see, with the time increases of the laser irradiation, the temperature dramatically increased, which indicating that there may be a photothermal therapy caused by these nanoparticles and the laser radiation. And then after the treatment, about 14 days, we measured the tumor volume of different groups. And as you can see here, the group which was treated with our nanoparticles and the laser, there is obviously no tumor growth again in 14 days. And compared to the control groups, this is a very exciting results that indicated that our nanoparticles can kill cancer and inhibit the tumor growth in vivo. So here is a summary of the tumor weight after 14 days. And as you can see here, the group with our nanoparticles treated with laser, most of the tumor has been treated. So in conclusion, in this study, in this project, we developed a semiconducting polymer nanoparticles that can carry the ions and the glucose oxides to perform multimodal therapy for breast cancer treatment in vitro and in vivo. And in the future, we're going to establishing a PDX model from by collaborating with doctors in the samples, and then we will establish a PDX model and investigate the therapeutic efficiency of our nanoparticles in that PDX model. So with this project finished, we would like to acknowledge the supporting from the University of South Dakota, which gave me the startup package to start my lab and start this project. And also I want to thank the NIH project, DaCCoTA project that's supporting this experiment. So that's all my presentation today. Thank you so much for your time.